First, I'd like to welcome Dr. Yoshinori Ohsumi, the 2015 Canada Gairdner International Laureate from the Tokyo Institute of Technology in Yokohama, Japan. He was given the award for pioneering the molecular elucidation of autophagy, an essential intercellular degradation system, and when disordered is linked to many diseases, including neurodegeneration, cancer, and infection. He was the first person to visually observe the function of autophagy, whereby cells clean up garbage within them and also kill potential invaders and ultimately keeping cells healthy. It works as a cellular recycling system to maintain homeostasis within the body. He went on to clarify the underlying mechanisms of autophagy, as well as all the specific genes involved in this process. So please join me in welcoming Dr. Ohsumi to the stage for his lecture entitled Lessons from Yeast, Molecular Machinery of Autophagy.
Good morning, everybody. Since I'm the first speaker of this symposium, I would like to start by sincere thanks to the Gairdner Foundation, especially John Dax and nominator selection committee of this prestigious award. Okay, and I also want to say thanks to the all member of my former lab member. Okay, today I would like to try to say what kind of thing we are doing and what's in the future in our study. Okay, I was born in 1945, and I started the yeast work in 1977, and that the talk I will today is started in 1988. It's already 27 years ago. We are working on autophagy in yeast more than 27 years. My organism I am using is a Saccharomyces cerevisiae, a small microorganism shown here. Yeast is very good model system to work with because genetics is very useful technique for this organisms. And when I started the yeast work in 1977, I started work on bacchio. At that moment, nobody interested, almost not so many people interested in this organ because they thought that just a garbage dump in the cell. But I thought this organ should have several important functions. And I started the biochemical analysis of this membrane, and we found yeast bacchio has various kind of active transport system, and B-type ATP is very important intercellular membrane acidification machinery on this membrane for the first time. And so bacchio is not the garbage. It's a very important organ to maintain the homeostasis of the cytoplasm. Then I had my own lab in 1988. I switched to my work to the lytic function of this compartment because I was interested in so much in the dynamics of the protein in the cytosol. So I started to work on the lytic function of bacchio because, as I said, bacchio is an acidic compartment, and it was known there was various kind of hydrolytic enzyme inside. So this must be homologous to the lysosomes in mammalian cell. Before I go to my own work, just let me say about the membrane dynamics of our cell. When I was teaching undergraduate student at Tokyo University, I always started this simple question: how many red blood cells are made in just 1 second in our body? Okay, it's a quite simple calculation. We can get the answer. 1 times 10 to the 6, 3 million cells per second. And how about hemoglobin inside the red blood cell? Calculation, we can get the answer. 1 times 10 to the 15th hemoglobin molecule per second. That means exactly the same amount of protein and red blood cell breakdown. So we adjust the equilibrium state of the synthesis and the degradation. Another example, our body making about 200 to 300 grams of protein every day, but we are taking in about 70 or 80 grams of protein. So for amino acid source, just these simple figures suggest that our new protein synthesis is maintained by our own protein degradation. So most of the amino acid for synthesis comes from our own protein degradation. So our protein is so nicely recycling. So degradation is important to understand. And okay, life is just maintained as an equilibrium state of the synthesis and degradation protein. And we can imagine 2 or 3 months, our almost all protein are replaced in our body. And so degradation is very important for survival upon starvation. And we know now degradation is not passive process. It is quite active and takes lots of genes are required for degradation, and energy also required. The intracellular protein degradation, the first landmark was done by Christian Dudube. In 1955, Christian Dudube found the lysosome as a lytic compartment in mammalian cell. Since then, Rockefeller Group analyzed by EM how to intracellular protein get into the vacuole, the lysosome. And 1962, Christian Dudube coined autophagy as a cytoplasmic material get into the vacuole to be degraded. Since then, not so much progress was done about molecular mechanism of autophagy, because degradation is not so easy to quantify, because if we compare the synthesis, degradation is not sensitive to measure, and we don't have good marker, and always EM analysis were required to get how autophagy is functioning. So it was not good system for molecular biologists. So it was required absolutely some good model system. As I said, I was working on yeast vacuole, and so the question I had is how and what and when cytosolic materials get into vacuole across the vacuole membrane. That's the question I had. Okay. So first thing I thought is when bulk protein degradation occurs in the cell, as you know, nitrogen starvation induces this kind of dramatic change, remodeling of the cell. Each cell becomes 4 spores. So this cellular remodeling is induced by nitrogen starvation. That means probably lots of protein degradation occurs in this process. Another thing is I always saw vacuole under light microscope. So vacuole is relatively large size, but nothing inside. So if I found some structure inside the vacuole, we can easily detect under light microscope. So under starvation condition, nitrogen starvation condition, we might expect some structure inside the vacuole. But if I use the wild type cell, nothing happened. So I wondered if I use the vacuole proteinase deficient mutant cell put into the nitrogen starvation medium, what's happened? Okay, this movie is the starting point of my whole work. I'm still working on this process, and this is a 3 hours starvation. You can see this is a real-time picture, and you can see many spherical bodies moving around in the vacuole. I thought this must be a quite interesting phenomenon. And this is the time course of the morphological change of the vacuole. After 30 minutes, you can see this spherical structure appear, and 2 hours, 3 hours, almost full of this spherical structure. We named autophagic body later. So we started the electron microscopic analysis of this phenomenon, and we found the structure inside the vacuole is a single membrane bound structure. And inside is exactly the same as the cytoplasm. You can see many cytosolic structure inside, and lots of ribosomes are taken up to this structure. And also many electron microscopy showed next to the vacuole, we can see this kind of membrane structure just end up in a portion of site, so still it's open here. And also double membrane structure surrounded. This is the autophagosome. It's already stick to the vacuole membrane. And this freeze fracture image shows the double membrane structure. Outer membrane is continuous to vacuole membrane, and inner membrane facing to the vacuolar sap. And this spherical structure is the autophagic body. And we couldn't see any intramembrane particle on this membrane. So autophagosome membrane is quite unique membrane structure. You can see outside several membrane particle here. So outer membrane and inner membrane is differentiated, and these are quite unique membrane structure. Okay, and you can see here the intermediate membrane structure, isolation membrane, and you can see mitochondria is taken up inside the autophagic body. So autophagic degradation is quite good for supramolecular structure degradation, such as ribosome, and even whole organella is taken up to this membrane structure, delayed. So autophagic degradation is quite good for degrade big structure like autophagosome organelle. So from those kind electron microscopic analysis, more than 20 years ago, we drew this kind of scheme of the autophagic processing is when each cell faces the various kinds of starvation, not only nitrogen starvation, suddenly this kind of small membrane appear and upper portion of cytosol form a double membrane structure, autophagosome. Autophagosome target to the vacuole, and fusion event occur, and inner membrane structure get into the vacuole. And in wild type cell, this structure is immediately disintegrated. So nobody could see this kind of structure. When I use the vacuole proteinase deficient mutant cell, we can follow the process of the autophagy progression as the accumulation of these autophagic bodies. So actually, this system has a quite good big advantage to work with under light microscope. And also, I want to say that vacuole is much bigger than rhizome, but membrane topology of this process is exactly the same as macroautophagy in mammalian cell. So this is a very good model for autophagy in mammalian cell. So we started a genetic approach to autophagy, and we didn't know anything about what the phenotype of the autophagy defect mutant. So we started just under microscope to get the autophagy defect mutant. Under starvation condition, the mutant cannot accumulate autophagic bodies. By this way, we got only one mutant, APG1-1. Now we call ATG1 gene. This was really autophagy defective mutant, but they grow quite normally in rich medium. But one phenotype we found is ATG1-1 mutant cannot survive. After 2 days, these autophagy ATG1 mutants start to die. So we speculate this loss of viability phenotype might caused by the defect in autophagy. Using this phenotype as a first screening, we could get 100 of autophagy defective mutant, and complementation analysis, we got 14 ATG genes from this first screening of autophagy defective mutant. Since then, we had 4 further ATG mutant by 2 hybrid analysis or interacting molecule. So the first screening was so efficient to get many mutants. So we started the cloning and characterized the ATG genes product. However, long time we could get any answer about the ATG genes, what they are doing, because sequence of the ATG genes said it's always it is novel gene and non-essential for growth, but essential for autophagy, that's all. However, when I was in National Institute of Biology, within several years, we could get many new findings. First, Nobuo Mizushima in my lab worked on the ATG12, and ATG12 was a unique ubiquitin-like molecule activated by ATG7, transferred to ATG10, and conjugate with ATG5, and combined by ATG7 for 16. Suddenly, 5 members of ATG genes was involved this quite unique reaction. Then soon, another interesting molecule was found, ATG8. This immuno-EM picture shows ATG8 reside on the isolation membrane of autophagosome membrane. So we thought this is a very good marker for membrane biogenesis of autophagosome membrane. So two graduate students work on this system, and we found ATG8 is another ubiquitin-like molecule and processed by ATG4 proteinase, and glycine exposed form is again activated by the same E1 enzyme, ATG7, transferred to ATG2, and to our surprise, ATG8 is not transferred to protein. It's conjugate with membrane phospholipid, phosphatidylethanol-amine. It's quite unique conjugation system, but those reactions are essential for autophagy. So we found among ATG genes, about half of the molecule involve these two kinds of conjugation system. And also we found autophagy-specific PI3 kinase are involved for membrane formation. ATG14 is a specific subunit of this PI3 kinase. And also we could see a product of the PI3P is actually involved in the autophagosome membrane formation. They get into the vacuole via autophagic body. So we found 18 ATG genes can subdivide into 6 functional groups: ATG1 kinase complex, and PI3 kinase, ATG2 18 complex, and one single membrane protein, ATG9, and 2 conjugation systems. And in my lab, Nobuo Mizushima and Dr.Yoshinori, he's here now. They worked on the mammalian side of the ATG gene, and first they found the conjugation system is well conserved in mammalian system. Now we know almost all ATG genes are well conserved to the mammal and plant cell. So ATG system has a long history evolution and in the evolution of eukaryotic cell. So I started the autophagy work here, and we found several interesting phenomena. One thing is, TOL is a, TOL is a negative regulator of autophagy. If you add rapamycin to the growing medium, autophagy nicely starts. But anyway, it took a long time to get the autophagy field become a so popular field. But once genes are found in mammalian cell, those identification of ATG genes change the landscape of autophagy work in. So many people join this field. Okay, once we have ATG8, Nobuo made LC3 that homolog of ATG8, and it expressed GFP tagged LC3 is expressed, and we can see now where and how much autophagy is induced in various tissue or even eggs. So that helped a lot to understand the autophagic process. And also knocking down or knockout of ATG genes showed autophagy is relevant for so many kinds of physiological events. Autophagy is not simply the recycling of their nutrient source. That is important for elimination of intracell harmful protein, damaged mitochondria, or even lysosome, and they function as elimination of invasive bacteria. And recently, many people working on how autophagy is related to the various kinds of disease in human. But our group is still working on ATG system in East because there are so many fundamental questions remain to be answered. So we started the localization study of ATG protein in the cell, and we found almost all ATG genes are at least partially localized the dot structure next to the vacuole shown here. We named that one as a pre-autophagosomal structure, the site of the autophagosome formation. So we thought this structure is important to understand. So we made every disruptant of one gene and what happened to recruitment of other ATG gene. And we found this kind of quite interesting hierarchical structure of ATG protein. ATG17 is an upstream factor, ATG113, and then ATG9, PI3 kinase, and then finally 2-8 complex, something like that. Okay, recently we developed a good camera system. ATG protein in yeast, it has less than 1,000 molecules per cell. And if this is ATG17, 17 means 17, 29, 31 trimeric dimer. So, but as you see here, ATG17 reside in the cytosol, vigorously moving by Brownian motion shown here. And the starvation condition, as I said, ATG17 localized the path. But as you see, path here and here and here, but still lots of ATG17 just moving around the cytosol. So a small portion of ATG17 functioning at the path. So how ATG protein are localized to the path? Recruitment of the ATG protein is important to understand. This is our working model. ATG under starvation condition, ATG7, I will show you later. ATG13 is a key molecule to start the autophagy, and then PI3, ATG9 vesicle comes and PI3 kinase join to the path. So hierarchical structure represents the timing of the recruitment of these ATG subcomplex of the ATG protein. And by collaborating with Inagaki and Noda, we solved many ATG gene structure and complex structure. Those helped a lot to understand what is the function of ATG protein. Now I will briefly mention ATG13. It's a quite important molecule for regulation. ATG13 has a unique structure, and terminal region is a globular domain, but C-terminal long structure is denatured structure, and so much phosphorylation site by TOC1 kinase complex. So this denatured region, we found ATG1, ATG13 binding site. And we found how ATG13 interact with ATG1 kinase. And we saw this is very important, and this is regulating phosphorylation dependent manner. So under starvation condition, the phosphorylated ATG1 tightly associated with ATG1. And also we found ATG17 interact with 13, just phosphorylation, dephosphorylation dependent manner. This serine residue form a hydrogen bond with ATG17. This is a key binding, key mechanism of binding. And this binding is required for the pus formation, as you see, and autophagic activity is gone. But we thought, okay, so we now know dephosphorylated 713 form this 5-member pentameric structure we call ATG1 complex. But it's not sufficient to induce autophagy. This structure should assemble furthermore 30 or 50. These complex should assemble. And what's the mechanism of this higher order assembly? And recently we found ATG13 has 2 ATG17 binding sites, and we could show ATG13 just bridge 2 ATG17 molecule. So this is a key finding, how high order structure is formed. And we recently found ATG13 C-terminal, just the C-terminal, and bind the TOC1 kinase. And TOC1 kinase complex recruitment binding here is also important for dephosphorylation state of the ATG13. And what the function of former? And we found this region is required for recruitment of ATG9 vesicle. That is an important process because this is the initiation of the membrane structure. We found ATG9 reside on the small membrane vesicle just moving around the cytosol, and it comes from Golgi, and under starvation condition, small number of ATG9 vesicle joined to the pus. So we want to know what is the, how ATG9 can recruit the pus. And recently we found former domain bind the ATG9 vesicles. And some mutant lose their binding to former domain mutant cannot recruit ATG9. So, and those mutants show the defective autophagy phenotype. So actually ATG13 interacts with so many ATG protein. So we believe ATG system is just a network of the gene, change their partner, changing the structure, during the autophagous membrane formation. So we need more careful analysis, what's going on in every step, what kind of modification is required to form a membrane structure. Okay, briefly I want to mention about the selectivity of the autophagy. Our machinery is just a core machinery for making a membrane. And if we add several unique protein, this system becomes a quite unique selective type of autophagy. For example, mitophagy requires some protein, hexophagy requires some protein. And also, one is a receptor, it's recognize a target, and some 11 is an adapter that recruit the core membrane machinery for making a vacuum, making an isolation membrane. And we identified various kind of a receptor, and also receptor is regulated by unique kinase phosphorylation we found. And recently we found ER or nuclear membrane is also the target of the selective autophagy in yeast. Okay, finally, I want to just say about the catabolism of the RNA by autophagy. Here you see if we introduce metabolome analysis, we found the transient increase of the nucleoside. That result from the RNA degradation within vacuole, because lots of RNA get into the vacuole, and that come to 3' XMP nucleoside, and nucleoside come out, and nucleoside become almost immediately to the base, and even under starvation condition, base secrete out from the cell. So RNA case degradation is not simply the reverse reaction of RNA synthesis. So we should more carefully analyze what's going on the metabolism during autophagy induction. So we are still lots of problems to answer, but anyway, this is our stage of understanding the autophagy in yeast, and those members was whole member involved in our analysis. I want to say thanks to all of them. Thank you for attention.